This is the last video for lecture 4, and I'm going to cover different quorum sensing systems. One way to think about this is that there is a very specific connection between the autoinducer and the receptor for it. So if you think about any receptor, any protein receptor, part of its job is to be very specific for what it can detect. And so the LuxR receptor very specifically detects that acylhomoserine lactone. But it turns out different bacteria can make different homoserine lactones, and they each have a slightly modified LuxR receptor for their own homoserine lactone. So you can have two different groups of bacteria right next to each other doing quorum sensing independent of each other because they have those specific LuxR receptors for their own specific autoinducer. Also, since this is happening in nature, some bacteria, they don't care about quorum sensing. They want to know whether they're surrounded by other bacteria. So they'll have a whole bunch of different LuxR receptors, but they don't have any other parts of the quorum sensing system. They are just listening for those quorum sensing signals from other bacteria for their own purposes. So this ends up being a really complex way that bacteria end up communicating. So that's what this slide shows, that there are different homoserine lactones with different acyl structures. So that's the length of the chain on the end of that molecule I showed you. But they're also completely different autoinducer molecules. So this one was discovered, I want to say around the year 2000, and it's super weird. It has a boron in it in this really bizarre structure. Anybody who's done a lot of organic chemistry and biochemistry would look at this and kind of shake their heads and think, how did this happen? But this is pretty common in nature. There are a lot of bacteria that use this exact molecule. And since many different species use this exact autoinducer, whenever they do quorum sensing, they will detect signals from other species that use this same thing. So instead of asking, am I surrounded by members of my species, they're asking, am I surrounded by bacteria that use this autoinducer, whether they're my species or a different species. So that's another surprising discovery about quorum sensing. So again, each pair of autoinducers and receptors is a language. And yeah, there are some species like Pseudomonas aeruginosa, which does those infections in people with cystic fibrosis. It has more than one quorum sensing system. It's trying to do really complex regulation of some of its genes. And that's all I'm going to really say about this quorum sensing, these alternate paths and the ones who listen. What I want to get into is a different quorum sensing system that we're going to look at in a little bit of detail. Partly because this is a system that is directly relevant to medicine, but also because it shows the effect of having a completely different type of autoinducer. So this is from Staphylococcus aureus. And what it's trying to figure out with its quorum sensing system is should it behave like a lone cell or a cell in a group? If it's by itself, it doesn't want to get swept away in the lymphatic system. It doesn't want to trigger inflammation. What it wants to do is adhere to whatever cell it's next to and just stay there and hide. But once there are enough of these cells, they want to start making toxins that let them kill cells and invade tissues. And they want to start releasing different factors that will prevent phagocytosis or interfere with immune responses. So instead of attaching or adhering to cells, they are secreting toxins and other factors. And that depends completely on whether they're alone or as part of a group. So this system is a little bit different from the LuxR, LuxI system, partly because the operon encodes both the enzyme that's going to make the autoinducer, so that would be like the LuxI of the previous system, but also the receptor, which is going to be in the membrane. So that would be sort of like the LuxR, but also there's a messenger protein and as part of a phosphorylation cascade, and that's AGRA. And instead of regulating mainly the amount of these, what they're trying to regulate is this RNA that's going to regulate a lot of other genes. So this is going to be a kind of a global regulator that's going to change the transcription of different genes throughout the cell. So instead of just trying to control how much of this operon is transcribed, this is going to go and control a lot of other operons that we aren't showing here. So let's see. So sorry, this is a duplicate slide. So this is the whole system showing what would happen if enough of this autoinducer peptide, AIP, autoinducer peptide, if enough of it is detected by this AGRC receptor protein, it will phosphorylate the AGRA messenger, and that will lead to increased transcription of both of these sets of genes. So then this operon will be transcribed more. You'll get more AGRD. You'll get more autoinducer. You'll also get more of this transport protein that pushes it out of the cell. You'll get more of the receptor that detects it. You'll get more of the messenger molecule. And so that gives you the feedback loop that ramps this up very quickly. But then the output that the cell really cares about is this, which is going to change the transcription of a lot of different genes. A useful thing you can do is compare this system to the LuxI, LuxR system. What do they have in common, and what is different between them? Definitely, they both have autoinducer molecules. They both have a possible positive feedback loop where if there's enough of this autoinducer peptide made by this gene, or the product of this gene, it will lead to more production of this gene and more production of autoinducer peptide. So that can be a feedback loop. And it's trying to tell whether the cell is alone or in a group. So the same thing happens when there are a lot of cells together. This concentration goes way up to the point where it gets detected. And if the cell is alone, this just diffuses away and the cell can't detect it with its receptor. But what's different? If you go back and look at the LuxI, LuxR system, there was no transport protein and there was no messenger and no membrane-bound receptor. The LuxI system had a completely different autoinducer. The LuxI autoinducer can diffuse across the membrane. So in the LuxR system, it doesn't need this transporter and it doesn't need a membrane-bound receptor because the autoinducer can freely move out or in depending on the concentration gradient. And that's what this slide says. And so that's again, another example of how the chemistry affects the biology. And you would have seen something similar to this in different types of hormones. Some hormones can cross a membrane and go into the cytoplasm and into the nucleus, and others can't. And it's based on the chemistry of that specific hormone. And in this case, the autoinducer, whether it's charged and cannot cross a membrane or uncharged and can freely diffuse across a membrane. That's the difference. This is a blank slide that I left in by mistake. So ignore it. So that's lecture 4. That's the end of unit 1. And that's the end of the unit where we are trying to understand everything we can about bacteria before we start applying what we know to the world. Topics coming up in lecture 5 is protists and fungi. So that's going to be both a short introduction to what protists and fungi are, and sort of where we see them in nature. But then the second part of that lecture is a list of protist and fungal pathogens. So you'll start to preview what's going to happen in Unit 4, where we learn a lot of different pathogens. And so you would study that material in a completely different way, and I'll walk you through that at the beginning of that lecture. And then lecture 6 is going to be viruses. Well, I will explain in detail what viruses are, and you have to completely change your thinking for viruses. They're completely different from bacteria and fungi and protists. So that will come up the week, that will come up after the Unit 1 exam. So good luck, and I will see you next time.